Good afternoon, all. Welcome you to this session. This time we have with us esteemed Dr. G.Hemaprabha. She is the principal scientist and head in charge, Crop Investigation Division, ICAR-Sugarcane Breeding Institute, Coimbatore. Ma'am is going to deliberate on the topic of uh, varietal scenario and need for modern approaches to develop sugarcane varieties. Ma'am, you are audible as well as visible, and your presentation is also visible. And we have started recording the session. You can start, ma'am.
Very good afternoon to all of you. Hope you had your lunch, all the trainees.
Yes.
Yeah, so the topic is on the varietal scenario and the need for modern approaches to develop sugarcane varieties. So this is actually a summary of the training program, especially with regard to the breeding aspect. That is about the varieties, that is a specific topic, and other modern approaches will be covered in detail in the subsequent lectures. So I am giving a preamble or an abstract of the training course. So, sugarcane scenario in the country, it's a very, very old crop. Right from the Vedic period, you have references in sugarcane, about sugarcane. And the real, from that is 400, references from 400 BC to 1900, that is 1900 AD. So during that time, many of the basic or the desi varieties or the basic species clones were grown. For example, in Assam and in southern India, it is mainly the officinarum or the thick forms of sugarcane, which is less in fiber and highly susceptible to diseases and climatic vagaries. And in the subtropical region of the country, as well as in China and other places, the Saccharum barbarae and Saccharum sinense, which are more fibrous and thinner types, and they are more climate resilient types. They are grown especially to withstand the severe summer and the harsh winter in those areas. So the real breeding in sugarcane started way back in 1893 by Walker and Cobbus. They are the pioneers, you can say, as far as sugarcane breeding is concerned. So the interest was to improve the sugarcane. So for that, they used this the thick forms of Saccharum officinarum mainly in their program. But this one, this Walker and this Cobbus, they took two other wild types, namely the Casor type or the wild, semi-wild spontaneum, officinarum spontaneum natural hybrid, and also by Chunni, that is the Barbara type. And they made some hybrids, and with that, all the sugarcane people would have heard about this name, POJ 2878. That is one of the progenitors of several varieties developed in all the sugarcane cultivating countries or sugarcane breeding countries in the world. So after that, in India, the major discovery or the finding or the development in breeding was made in 1918, actually, when the institute was started in 1912 by the Britishers. That's Britishers only identified Coimbatore as a place for sugarcane breeding way back in 1912 because it is endowed with a natural flowering and seed set, which is essential for any breeding activity. So sugarcane breeding place was found to be Coimbatore, and still it enjoys to be the only or the major sugarcane breeding center in the country. Of course, there are some places like VSI Pune has a place, then some private companies also, they also have some breeding program. But still, no, it is sugarcane breeding is mainly concentrated in Coimbatore because of this flowering. And this was identified by this location was identified way back in 1912 by the Britishers. So they, out of just mere curiosity, took a wild grassy type of called spontaneum, and they used in hybridization, and to their success, to the amount about some 86 seedlings only they produced in the first batch, and from that they could identify 27 cocaines, of which 205 became very famous. And that is the sweet revolution started way back in 1918 in India. And this was considered to be a major finding as well as sugarcane breeding in the world. and this technique of interspecific hybridization was copied by other countries, and this effort was subsequently carried out by Sir T.S. Venkatraman for his outstanding contribution with regard to development of several tri-specific hybrids, which were cultivated not only in India, in 24 sugarcane growing countries also. Even today, as Director was mentioning in the morning, some of the old types are still in cultivation, and all these owe to Sir T.S. Venkatraman. And because of his immense contribution to sugarcane agriculture, the Britishers knighted him, gifted him this title of Sir, and after Sir C.V. Raman, he is the only Indian scientist to be knighted by the Britisher. So you can imagine the contribution made by this gentleman as far as sugarcane breeding. So once the platform is set, it was very easy for the subsequent people to continue. Though sugarcane breeding is a tough process, the periodic development of varieties continued. And you can see this figure. This is the old barbarate Kata, and this other Kata is the form which was grown mainly in subtropical India. And this variety 205, that is green revolution or sweet revolution, demonstrated in the field itself. You can see the increase in height, increase in the plant structure, plant growth, etc. So showing that this is the real demonstration of green revolution started in India in 1918. This is in Punjab. This variety was released for Punjab, and subsequently it was taken to other parts, including Odisha, where it could withstand severe waterlogging conditions as well. So after that, during the 100 years or more than 100 years of sugarcane breeding, so after successful sugarcane breeding, after the first release, there are many varieties released in subtropical and tropical India. This number is not fixed; it is growing yearly, and this table, this is regard with the tropical states. You can see that during the first century of sugarcane improvement was only dominated by one variety. Soon you can see the number increasing. 213, after 205, 213 became a more versatile variety. Then the first wondercane of tropical India is named as Co-419. It's a which brought a very serious impact on sugarcane production in the peninsular India. This is tropical India. You can see it lived for more than 50 years or 60 years till it succumbed to red rot disease. And this was soon replaced by 6304, the second wondercane. That was in cultivation to 30 to 40 years, and mainly a variety for tropical India and especially for Tamil Nadu, characterized by high yield and moderate sucrose. And then came the third wondercane, that is called 86032. Even today there is no clear substitute to 86032 that enjoys the status as the wondercane of the present moment in tropical India. And similarly in subtropics, 205 occupied for about 20 years. Then 213, which became a national variety both in tropical India and subtropical India. Then 312, which is characterized by high yield, that came, which occupied more than 40 years. Then came, this is called as it's a wonder variety, that is 1148. That was still cultivation till the beginning of the 2010. Many, with more than 60 years, this variety, or 60, 70 years, this variety ruled the subtropical belt. And against US 767 was another variety even now in some areas 767 is cultivated. So you can see some varieties come and go, and some varieties have a long life in the field. So we have to find out the reasons why some varieties are having a greater or a larger field life. So now India is divided into 4 major zones. That the peninsular zone is the largest zone, comprising of the states of interior Tamil Nadu, Telangana, Karnataka, Maharashtra, Gujarat, Madhya Pradesh, Chhattisgarh, Kerala, and also some part of Andhra also. Now that is interior, that is the Chittoor region and the near Vijayawada. That region is also under peninsular zone. So this is characterized by high productive zone where there is a congenial or a conducive environment for sugarcane to grow. But of late, drought and water stress are the major stresses in this area. Coupled with salinity, the major biotic stress is smut. Red rot is not a very major disease in this region, but it is prevalent in the Gujarat state. and also in the Tamil Nadu, the coastal region of Tamil Nadu, which does not come under this zone. So this is an area where you can achieve more from sugarcane. So during the past, that is from 2000 onwards till 2020, you can see a host of varieties have been released for the peninsular zone. But you can say after 86032, which was officially released in 2000, it completed its testing in 1994, and still there is no clear substitute to that variety, though the variety is being balanced by a few other varieties developed from Coimbatore as well as from Maharashtra, especially VSI Pune and Padagon centers. Now, this is an important variety. I think everybody working in sugarcane is very, very familiar with this variety. It is a 32, a very durable variety, a very attractive variety, and which has sustained sugarcane cultivation in the peninsular region. And you can see it occupied more than 1.1 million hectare in the country. Now the yield is almost stabilized at around 0.85 million hectare. It's more than 80% of the area in Tamil Nadu. It was 90% in other states. It was around 70% area by this variety. Now the area is slowly being maintained or reduced. 80% in Tamil Nadu and 65% in other states is occupied. In southern states is occupied by this variety. So with the cultivation of this a very, very versatile variety, which is amenable to any mechanical operations. It is amenable to wide row spacing, narrow spacing, or any spacing. It can be harvested at 12 months. The variety retains its high quality up to 16 months. So all advantages for this variety, good return yield it gives, but there are a few minus points, like it is not suitable for places having more of waterlogging. It is susceptible to internode borer. And especially nowadays, when mechanization is becoming popular, just for mechanization, the variety should have a very deep root system. So this does not have a very deep root system, though it is good for mechanical harvesting. So deep root system varieties are more successful under mechanization. So, and also, you have that's just by you will get confused hearing the names of varieties. I want to make it very clear, and it's a very, very simple terminology as far as sugarcane is concerned, and especially for co-varieties. The first two digits denote the year of identification of that variety. For example, 86032 was identified in the year 1986. And these are the rest of the culture numbers. That is 32nd entry. Like that. So it is very easy to remember the varietal names once you know how a variety is named. So now this is a very huge variety which brought immense benefit to the industry as well as to the farmers. It had a higher yield, maintained around 105 tons per hectare, except during the drought years, the yield was slightly low. And as a recorded highest yield 324, now even 400 tons are also achieved by some of the achieving farmers. And that the number of that is 100-ton club, that is 100 tons per acre club. That's a group of farmers having this 100 ton. That number is increasing because the variety is the same. You have to use a rejuvenated or a good seed material coupled with good agronomic practices. You can always aim for a 100-ton club. This is one of the ways by which doubling of farmers' income can be achieved. So many of you are from extension disciplines and from KVKs. It is your take-home message that is all the good and effective production technology should be translated to the farmers or the end users through different modes of conveying this message to the end users and farmers. So now, for example, this particular variety over years, it has brought a huge revenue, 203,546 million rupees to the exchequer, both for the cane, for the factory people as well as for the farmers. Farmers gained by the increase in the yield, and also factories achieved because there was a 0.5 to 1 unit increase in sucrose recovery compared to the earlier varieties. So this was a win-win situation to both the farmers as well as millers. And that is one of the reasons why still 86032 is under cultivation even after 40 years of its identification. Then two other varieties which occupy a larger area in peninsular India, COM 0265. About 4 or 5 years back, this was a variety which was spreading very fast in Maharashtra because of its high drought tolerance and saline tolerant condition. And it is suitable for Atsali planting that for 18 months. Harvesting at a, though it's a moderate sucrose variety, when it has harvested in 18 months, that's a very common practice in Maharashtra. But now this disease, there's some diseases like brown spot and other things are happening in this variety, and the area is coming down. And another variety which was released, which was tested in ICRIP and identified by Kolhapur Center as well as in Maharashtra and Kolhapur Center and also in Karnataka is 92005 because of the excellent jaggery it makes. The jaggery from this variety is fetching an extra price, at least 1 rupee per kg. In some cases, it is 2.5 rupees to 5 rupees per kg. This variety, the jaggery of this variety is fetching. The highly very suitable variety and very attractive variety, which is widely grown in Kolhapur, and now it is spreading to Karnataka. And even not only for jaggery, now around factories, 32% of the area in sugar factories in Kolhapur is covered by this variety because the sugar recovery from this variety is also much higher. So it is now spreading to northern Karnataka as well, especially in places where there is no problem for irrigation. Now another variety, because now the future varieties, it should not be like cross something into something to get something. You should have some wild blood, some new germplasm incorporated, because now climate resilience is becoming a major part for the success of a sugarcane variety. So we have to incorporate some wild blood. At the same time, we have to keep the yield and quality. It's a very, very challenging task. Sugarcane breeding is a very challenging task. So many aspects we have to take into consideration. For us, for breeders, sugarcane should flower. For industry, it should not flower. Farmers, it should not flower. So it's a very tricky way of choosing a correct parental combination. So it clicks sometimes, and again, we have to try, and through experience, it is easier, it becomes little more easy to breed sugarcane. So here it has some wild blood from a new source of officinarum, though it's not, officinarum is not wild. So with that, you can see it is more tolerant to red rot, smut disease. Generally, this type of behavior we don't get as for a good sugarcane variety. That is red rot resistance, smut resistance, tolerant to drought and tolerant to salinity. So this is a future variety probably. In Tamil Nadu it is tested. But thing is that when it is compared to 86032, yield is always good. It is very less for a flowering or fast flowering variety. And slightly, initially it was slightly better recovery, but now it is almost on par. And we have to test it in other states to identify its suitability. Then another variety which you know about, COC 671, is a major variety released in sugarcane for the highest sucrose content ever, which improved the sugar recovery in Tamil Nadu and other region. But it succumbed to red rot by the end of previous century, 20th century, became highly susceptible, and there was a immediate need for varietal replacement, and 86032 came at the right moment then. So the sucrose content of COC is much reflected in 09004, and it is resistant to red rot. So we thought we expected much from this variety, but now today morning Kavarda was telling there is flowering in this variety. But naturally, this is a high yield sucrose variety, no doubt. But and plant crop is also excellent. But we have to have a more of ratoon management to take up this variety. Then 06022, which I did not want to mention initially, but I find that many participants are from Maharashtra. So in this variety, though it is released for Tamil Nadu, highly drought tolerant variety. You cannot call drought tolerant, drought stress. Even under extra water logging condition as well as water stress condition, this variety is doing well. So recently we had some experiments in the Kolhapur region. So there this variety was found to excel. This topped the list. So that's why this variety is included. It's a very tall and erect cane with good cane population. But the flowering in this variety has to be noted. But otherwise, it can be a very good variety in many of the tropical region. So it is a fact that sugarcane breeding is not a breeder's job alone. We have to have a tie-up with the factories. By testing it in the factory region, then only we will know the real potential of a variety, whether a variety will succeed, what is the sugar recovery. Sugar recovery is not in the hands of a research station. For that, a sugar industry is required. So understanding all these things, when our director took charge as the director of this, Dr. Bakshiram took charge as the director of this organization. He, along with the industry, after knowing the pulse of the industry and after discussing with the industry, started a new program. It's a collaborative mode between the Sugarcane Breeding Institute and sugar factories in Tamil Nadu. So a test of around 20 varieties initially and 16 varieties at present are being tested. So the results of this program was a huge success. So with that, the first variety to be released in a very short time was called 11015. Actually, you know, because 11015 is a futuristic variety, not only for sugar production, but also for ethanol production because of its extremely high sucrose content. And not only sucrose content at 12th month or 10th month, even at 8th month, the quality is very good and the variety can be harvested at 8 months. So this is the first short duration variety released for commercial cultivation in sugarcane. We had some short duration varieties earlier, but nothing as good comparable as 11015 with a very good ratoon performance, very much amenable for harvesting at 10th, 8th month. So many of the factories in Tamil Nadu, a few factories in Tamil Nadu are now shifting to having 3 crops in 2 years. So that means, though this variety has a tendency to flower, or at least 40 to 50% flowering, by adjusting the planting time, the variety can be cultivated profitably, both for sugar production and also for ethanol production. And the farmers continue to get one more bonus crop in the course of 2 years. That's the third crop. So with the result, you know, by suitably adjusting the planting, the flowering period can be easily avoided, and the sugar flowering period, sugar flowering in this variety can be easily avoided. So this is a very promising variety. Now we have a demand from other regions, including up to Gujarat, and even in from UP and Bihar also factories have taken this variety. But of course, flowering is a problem in this variety. But as I told, don't be scared by the flowering. It can be easily handled or managed by adjusting the flowering time. So this is a way of doubling or at least improving the, at least some 30% improving the profitability of the farmers. It's a very fast growing variety and it responds very good to the fertilizer application. So with the result, it's a good ratooner, and even for jaggery, this is a very good variety with golden yellow color jaggery. And you can see how the maturity of this crop, even at the 8th month, the purity is 85%, which is more than 85%. 85 is the threshold to know the varietal maturity. And both CCS sucrose percent, CCS percent, cane yield, everything is high in this variety. So there are a host of other varieties. Morning again we heard that 8,000, that is 12121 of BSI. Then Dhanush 92020, then MS 10,001, and all these varieties are grown or were grown in sometime in the peninsular zone. And another variety is COV 09356. This is from Biur, which is not in our zone, which is under the East Coast zone. Even that variety is now occupying sizable area in Tamil Nadu, where waterlogging is a problem. So now coming to the East Coast zone, here the problems are mainly waterlogging, snapping, lodging of cane, and red rot incidence in sugarcane. So all the varieties bred for this region should have resistance or tolerance to these factors, because this is an area you get now, again there is a warning of cyclone. So cyclonic winds can snap or even uproot the canes. So that is the reason why this region, the cane cultivation is more challenging. So though many varieties have been released, right from 86 to 49, then 06030 and the Kadlur variety 13131 that's recently released. There are many varieties released for this region. And also for Maharashtra, Andhra, there are 3 research stations, one at Anakapalli, one at Biyur, and then there is a Rudrur, or Rudrur comes under Telangana. Those varieties of Rudrur are also cultivated in this region. And also we have a center in Panipoila, that is in Odisha. So, and also Kadalur and Elikupam are other centers. So it's in the coastal region. So many should have waterlogging resistance, and both early varieties are much preferred to complete the planting and harvest quickly. And some of the 03, 13, 339, 039, 936. Then some of the varieties from Tamil Nadu Agriculture Universities are all grown in this region, and also Odisha varieties. The sugarcane, there are 2 factors in sugarcane. One is a broad acceptability or adaptability, that's the zonal varieties. And also there are some varieties which are suitable only for specific regions, very specific regions where like tannery influence or in waterlogging area or in red rot plus other factors. So in that one, no, we have to have a thorough adaptive trial testing to know which variety is suitable for the specific region. So researchers or breeders sitting in Coimbatore or in a particular place cannot identify varieties for the different areas. For this, there should be a good cooperation between the research station and KVK or even sugar factory to identify location-specific varieties. So North India, subtropical India, there are 2 major zones. One is the northwest zone, and another is the north central and northeastern zone. Now they are merged together as a single zone. So northwest zone is having Haryana, Punjab, Rajasthan, Uttarakhand, and Western UP. Red rot, wilt are major problems. Drought, salinity, high temperature during summer months, cold during winter, and the varieties suitable for June planting, that is after the harvest of wheat crop. These are the characters which are needed for this region. So many varieties. There are some very vibrant research stations apart from our Karnal Center, like Shahjahanpur, Pantnagar, JB Pandit University, Uchani Station, then Jalandhar, Punjab stations, and then our own sister institute at Lucknow, then Kota, Rajasthan. So many research stations are working in collaboration with SBI by making crosses at Coimbatore through the plug supply program. So, but only a few varieties are now penetrating. A typical success is the 238. I think now everybody will be familiar with this variety, especially after Dr. T.R. Sharma's inspirational inaugural address. So this particular variety is a wonder variety occupying more than 54% area under sugarcane in the country. You can imagine one single variety occupying 54% and in 85% area in the subtropical region. It's a huge success. Now it is around more than 25 lakh hectare by the single variety using 1920. And this is a single example of how a variety can change the entire lifestyle of people. Here in northern India, though it is released only for these states mentioned along, it has spread to Bihar, and even it has come to Chhattisgarh and slowly going to Gujarat and even up to Odisha. So this variety has changed the lives of many, even more than 2-3 lakhs of farmers. And each farmer, they are getting a benefit of totally during this 5 years, got a benefit of 65,000 million rupees. And per hectare itself, they are getting around 40,000 rupees extra by cultivating this variety by way of its high yield and high sucrose. Sucrose much better than the earlier varieties. With that, the factories are also getting more than 1 to 1.5 units higher sucrose. So this is a typical example how a variety can change the entire scenario. Because Maharashtra, Karnataka, or the peninsular India was considered to be the cradle of sugarcane, where sugarcane yields are high. But everything has now been turned upside down by a single variety. the 238 that has become subtropical India as the major sugarcane belt in the country. The subtropical India is now crippled with other problems of drought and problems of water scarcity, water surplus in area. This is a place where water is also abundantly available, and this variety is becoming a very, but only problem is that of late this variety is now succumbing to red rot. Especially red rot can be controlled to some extent by adopting a clean cultivation to a tissue culture derived planting, using tissue culture derived planting material. So you can see it is in this is in Madhya Pradesh, in Bihar, and all the states. You can see how they are doing cropping in sugarcane. You see the height of a Punjab farmer is more than 6 feet. You can see the crop growth there in Punjab in very attractive crop, and it's only seeing to believe. If I say now the crop is so high, especially to a person sitting in south, it is not possible. You have to see and believe the height and the growth of this crop in subtropical region. So you know that as 238 is slowly being eliminated in the picture, though it will be very difficult to have a replacement to 238, a variety which is being highlighted is 118, which was released along with 238. Its sucrose content is much better, and it is a thicker variety, like a tropical variety. And this variety is suitable for autumn planting, and this variety is now considered as an alternative. And recently another variety called 15023 has been identified. So since the release proposal, though it is unofficially known that this variety is released, since the official launch has not happened, I'm not covering much on this variety. This is actually a lot of expected from this variety, a red rot resistant variety with high sugar, which is now being covered, especially in Punjab region. So another variety which is a climate resilient variety with high fiber and suitable for cogeneration, suitable for water stress and waterlogging condition is 98014. This came much before 238 and 118. And here, because of this advantageous condition, it is grown in some area. And several varieties have been released, including the Punjab variety and also the COLK 94184. This is a high sugar variety. Though this variety is not released for this zone, this is cultivated widely in UP. And also some of the north that is Bihar varieties have also entered into this region, apart from Kokpand, that is from Pantnagar, from Uttarakhand. And now coming to the north central zone and northeast zone, this is apart from the problems of northwest zone. This zone is characterized by early drought, late water logging, drought, wilt, borers, and other problems. So this is mainly comprising of the states of Bihar, central and eastern UP, West Bengal, and northeastern region. Here also many varieties have been released over years from 2000 up to 2019. And among these, the variety that Birendra plus COLK 14184 is a very important variety for this row zone. Not only for, as I told earlier, it has now gone in UP, Bihar, and West Bengal. And once the area covered was 2.5 lakhs, now it is not slightly less. And there are other varieties released from the Sivarahi Center of UP Council of Sugarcane Research, from the Shah Jahan Center. There are a few varieties available. And also Burlixon in Assam. So this region is a very different region. The good varieties of these plains will not be suitable in this area, and vice versa. So they have their own varieties. And also some of the regionally developed varieties are grown there. So now coming to the lifespan, some varieties live for a more period. For example, these are some of the varieties which are having high field life, whereas some of the varieties go away quickly. The main reason is the red rot breakdown. Red rot breakdown is faster in our experience, though it is not a confirmed thing. It is faster in high sugar varieties, as you saw in COJ 64 and COJ 671. So there are some reasons for the pathological, that pathogen specialization, pathogen modification, and with the result, the variety become vulnerable and susceptible. So So now, after the success of 238, subtropical India has gained a lot from cane yield. Earlier, it was 65 tons. Now the average yield of farm of UP also has increased more by more than 10 tons per hectare, and it was possible. There are some achieving farmers in this region who have achieved 325 tons. And though these are all much above the theoretical possible limit, it is possible. That is one of the major themes of this training program. Why some farmers are getting, why not others? So that means it is possible. Varietal potential is there. By following suitable agronomy, suitable management practices, we have to get higher yield. This yield gap has to be narrowed, and the variety has to perform much better, like towards the physiological limit of a variety. For this, there should be a clear or a perfect understanding between the breeders, agronomists, and the sugar factory personnel, along with the farmers and the extension workers. So this is a very important aspect of this training program, how we can increase the number of achieving farmers, through which it's a very, because sugarcane, that is just, there is no need to just toil in the field. Now everything can be mechanized by adopting a wide, this should be covered in the subsequent lectures. It makes more scientific and more interesting and more challenging for the youngsters or the unemployed youth to take up to this, using this machinery, using a complete mechanized operation. It becomes more scientific and more enthusiastic and more rewarding to cultivate sugarcane. So please carry this in your mind. And now this is the one part. The next part is the summary of the, or the abstract of the lectures which will be following. Why you need improved approaches and the ways to develop sugarcane varieties. You know, sugarcane breeding is not, as Dediji was telling, it is not a simple joke. If you start your crossing now, you get your product after 14 or 15 years. So that means a scientist who has just joined will be able to get first year of, if he is lucky enough to make crosses in the first year, probably after 15 years he will get his first product as his own. Through association, that is different, but by your own. So that is the challenge in sugarcane breeding. It needs so many years of testing, continuous testing in different stages, in small plots, in slowly increasing from a single clump or single seedling to a larger area, and finally you give it a co-number or a culture number from a station. Again, it has to enter national testing in different regions of a particular zone through under All India Coordinated Research Project. For that, again, it takes around 5 years for completing, including multiplication, and finally you get a variety. So when finally you get a variety after this, the condition or the environmental condition may be totally different. That time we would have screened for red rot or some disease at this stage. Finally, when you come, you find a newer challenge, newer pest, newer disease, so which cannot be easily addressed. So this is a very important, very tricky and a very complicated or a frustrating part for a sugarcane breeder. But of course, we have to face the challenge. So it's a very, very challenging and tough task of breeding a sugarcane variety or developing a sugarcane variety. So the strategies also, industrial demands also vary. Earlier, for example, you had 2.5 to 3 feet spacing. Now it should be 4, 4.5, 5, 6, 10 feet spacing, which should be suitable for intercropping. And like that, it should be capable of harvest at 8th month, 10th month, sometimes 18 months. There should not be any decay. Sugarcane should not flower. So all these challenges are borne in mind when a breeder is making a cross. So also there is need for multi-trait genetic stocks, because that is to serve as parents for different stress conditions. Then again for climate resilience. Then genomic, gene identification, etc., by which sugarcane breeding can be more scientific and more success-oriented, and transgenics, by which a defect can be rectified or a new value can be added, and genome-edited sugarcane. They are all considered as the future prospects in sugarcane breeding. And over the years, I should say that it has been a success, or it has been highly rewarding to see that earlier, I think the year is missing here. This is during the early period. Then the types with less than 15%. It's only very high. I think here this chart is not complete. And red rot resistance and sucrose. That I mean to say that to combine red rot resistance and sucrose, the proportion has increased over decades. And presently we are able to get types with more than 22% sucrose with high amount of resistance to red rot. So one thing we have achieved, it is easier to combine bricks with high sucrose and red rot. Over years we have come up to that confidence. And now for the future again there are problems. Then we have to study the per se performance. Then crossing is generally guided by the per se performance, how that particular variety of the parent is, whether it is resistant, whether it is having high yield, quality, etc. Then it should be a good combiner. It should be able to combine with another parent to give a good progeny. For that, we have pedigree method or molecular diversity methods are used to identify little more diverse parents in crossing. Hello? And then other methods are also, so that molecular methods are widely used, and by this classification we can identify, we can pinpoint or pinpoint or pick some of the good varieties from different groups, so that it has to be to exploit heterosis. And then another important challenge of the present day is to have short duration varieties. This is not only for, you can see the sucrose up to, the bricks up to 27% at 8th month. That is the potential of that newly released variety. So why you need short duration varieties? Namely, because sugar recovery is very low during the start of a season and the end of a season. So this brings down the average sugar recovery of a factory. So we have to improve at the early season and the late season crushing season. And now sugarcane is emerging as a bioethanol crop. You have to produce ethanol. Ethanol cannot be confined, that the production cannot be confined only during the crushing season or the harvest season. You have to have round the year ethanol production, or for that short duration varieties the only way, which are amenable for harvest, planting at any time and harvest at any time. And if you have to have 3 crops from 2 years, we should be able to grow them in special season and main season. And there should be a very good ratooner, because one plant crop is followed by 2 ratoons. You should have climate resilience. It should be wide row planting, amenable to wide row planting as well as for mechanization. And in the course, we have identified several short duration varieties combining high sucrose. You can see the sucrose more than 20%, and also the red rot resistance. And that is one aspect. And another aspect is to have multi-trait genetic stocks. Sugarcane germplasm. India is a wealth of sugarcane, has a wealth of germplasm, and we at SBI are maintaining a big collection of germplasm. So this germplasm has to be maintained, evaluated, documented, catalogued, and utilized in breeding program. Actually, if you see the rate of utilization of germplasm, it is very less, and that proportion has to be increased. And there are several that is mobilized form or improved intergeneric and interspecific hybrids with us, which are the future donors for the future stresses of contributed by climate change. And another way of we are achieving is through developing a multi-parent advanced generation intercross or magic population. Here we are pulling 8 different genomes. That is from officinarum, also officinarum, that is from Saccharum officinarum, Saccharum robustum, Saccharum spontaneum, Saccharum barbari, and sinensis, Arianthus arundinaceus, bengalensis, and procerus. So we are in a very scientifically, we are pooling and using molecular markers. We are identifying the true hybrids. That is by making into 2-way, that is 2 genomes are pooled to make 2-way cross. And 2-way cross hybrids are again crossed to make 4-way, and 4-way to 8-way, which represent the genome of 8 species, and which we expect. Now we have come up to this stage, and the initial materials of 8-way are being developed. Expect they will be more elastic. That means they will be more climate resilient to address to the future needs. We have carefully chosen the parents, so that it's already some amount of stress is incorporated in the parent. That is some of the derivatives at each stage are taken. So this is a major aspect which we expect. And another thing is that, only officinarum, that thick type is alone used as female. So using this wild types as male, that is a new novel genetic source with for cytoplasm, totally novel types. So we have developed so many variety clones in this process using cytoplasmic diverse lines, and one such clone has entered into our ICAR system, and now that has completed last year, where this is identified to be a very high yielding type, but the sucrose content was not sufficient enough for its release, and we found 4 Erianthus chromosomes in this improved hybrid. Again, we are using that in breeding program to improve its sucrose content. And in the process, now you can see how different genetic material are developed with Erianthus proceres with improved officinarum, robustum with Erianthus arundinaceum, sugarcane sorghum. Similarly, many such hybrids have been identified if you are registered with the National Bureau of Plant Genetic Resources in Delhi. And again, we have developed more. And a way of improving the precision in breeding is through marker-assisted selection. Especially what we are doing is that for hard to select characters like drought, which you cannot screen during in a seedling stage, or drought tolerance. So our earlier studies have given rise to several candidate gene markers, which we are initially using to identify the resistant types. This project has been initiated very recently, and hopefully we'll be able to indirectly select for drought resistance and drought tolerance. And for that, several candidate genes have been validated using qRT-PCR as well, which we are using as functional or more useful markers in our marker-assisted selection program. And sugarcane is a crop which can develop several genes can be identified from the wild germplasm. Of course, our team has identified several genes as well as promoters. And this is especially in regard to one pathway. Especially we are concentrating on drought. Several drought responsive genes have been identified. This is just one example. This will be covered in detail in the subsequent lecture. So glyoxylase pathway, there is GLY3 gene, which is with a single gene is sufficient to incorporate this particular character. So this gene, several transgenics have been developed, GLY3 types, and they are now in the glasshouse and awaiting a limited field trial. These are the transgenic sugarcane with improved drought tolerance compared using 86032 in genetic transformation. So this is also progressing well. And in agrobacterium mediated transformation is also being done using our own promoter that is identified from a wild rice, and also we have a promoter from sugarcane itself, from Erianthus and the sugarcane. And some of the events have been characterized for physiological parameters to measure their tolerance to drought using some of the very specific drought tolerance indices, physiological indices. For example, cell membrane. This is the uncontrolled plant, and you can see the transgenics representing less cell injury. So the transgenic having less cell injury, similarly for other parameters. And here also many are now awaiting, that the southern confirmed plants are awaiting field testing, field trials, limited field trials. And apart from sugarcane, we have canes or energy canes, which are with very high biomass. If you see the picture here, you can see very huge biomass, which does not require any management. It will grow by its own. These are some of the Erianthus derived hybrids which are grown for bioethanol production, bioenergy production, and a host of other materials. Now that is for, they can be used in cogeneration ethanol. That is through that is not the primary ethanol, from secondary ethanol from bagasse. That conversion, that is bagasse based ethanol production. And they can see the biomass, the huge biomass with dry matter itself is more than 100 tons per hectare. By dry biomass. So with this, there are many, even textiles are made with the sugarcane bagasse, sugarcane fibers, Then it's a very good material for particle bomb, particle board, then for plywood production, plywood manufacture. Then it can be this bagasse cup, not only for sugarcane and also for the energy cane, can be used for the platform for mushroom cultivation, for cutleries. Even Ganesha during our Ganesh festival with the sugarcane bagasse was prepared. And similarly, a host of material can be developed, can be manufactured from sugarcane, which can be taken as a profession by the self-help group or some of the startup companies, startup groups of youngsters or even ladies, housewives can start this to bring extra energy or to double or to improve some of their improve their monthly income through sugarcane. It can be done in a collective manner. Again, I plead, I request the KVK team to take up such activities for the benefit of our women folk in the rural region. Then another aspect which I would like to cover is that now you know the sugarcane cultivation, the cost of sugarcane cultivation, one of the major factors is cane cost. That is cost of the cane, seed material. About 8 tons of cane is required to plant 1 hectare. So now, you know, we are using the cane cuttings. Now, sugarcane flowers. And the flower, this the single flower is called the fluff. From that, we produce the flower. That is the breeding method. So once, why can't you use this flower or the single seed as the sugarcane propagate? So this project was conceived a few years back, and we are now progressing in this way. But you know, by taking the fluff of one sugarcane flower, you find a lot of variability because it's a highly polyploid and heterozygous material where the variability is so large that you won't be able to replicate a parent. The progeny will be totally different. So for that, you have to improve the homozygosity. You have to derive seedlings as good or looking like begets like. That is a term of genetics. For that, it should be genetically pure. We have to improve the genetic purity. That is, or improving the homozygosity. We are trying many methods, and one method is repeated selfing. So by this method, now from 6 to 8 ton seed can be reduced just to 30-35 grams, which you can simply take in a cover, in a pocket, put in a pocket and take for sowing in a field. For 1 hectare, this much is required, but for that, a lot of research has to be done to make that true seed really true to the parent. So for that, our work is in progress. So once this is developed, sugarcane becomes a very, very easy crop. New varieties can be easily be taken, distributed, and the seed exchange is easy, seed varietal replacement is easy, and sugarcane productivity also, that is cost of production will be drastically different. So for that, we are doing with the aid of molecular markers, identifying a very near homozygous line. And to some extent, we are able to develop some near with less aggregating population. I think we can say we have achieved only 10% towards the road to success, on our way to success. But anyway, we'll be attempting it to get to this. Once it is standardized, then the cost of cultivation of sugarcane becomes very easy, and it becomes a fantasy for everybody, because the new variety can easily be supplied to the end users, and the varietal replacement becomes easy. Disease and pest control can be controlled, because you know, when the sugarcane it is grown over generations from repeated clonal propagation, and it accumulates viruses. With the result, many leafy foliage diseases appear. This is just from the seed, so it will be very healthy and it will be green, and it will be free of diseases. So in this way, the yield levels also will be higher. So now another is so the virus-free the tissue culture. That is again that method. We are able to now supply sugarcane seed, and also for the varietal replacement. Now our capacity has been increased to more than 1 lakh plants can be produced, and with the result, you know, that we get more healthy, rejuvenated method for sugarcane cultivation. So now, coming to the conclusion, the potential of selecting better yielding varieties is now for short duration early and mid-late varieties have increased over years. And genetic base broadening of hitherto unutilized genetic sources is very much essential. Even DDG was also mentioning this is very much essential to address the new and emerging stresses. And we have a very strong group working in this aspect, and the progress can be very good with regard to the utilization of several unutilized genotypes for different characters. Tissue culture based seed production has enabled rapid multiplication of healthy plantlets, and also to replace all the old diseased plants and to rejuvenate some of the old varieties, including 86032 is completely rejuvenated through this method. And True Seed offers a lot of potential to transform sugarcane agriculture. Molecular techniques are complementing sugarcane improvement. Though we have not come to an age of using them directly, to some extent we have succeeded to use gene specific and SNP markers in our SNP markers are being developed. And then gene identification, characterization, and functional validation and transgenic development. And this all will lead to the development of improved varieties to meet the future challenges. So you can see this is a rejuvenated sugarcane. You can see the yield. This is the way of doubling farmers' income. So through a use and collective farming and the correct suitable rejuvenated or a healthy seed followed by different cultural operations will definitely make sugarcane more profitable and remunerative in the coming. And this is a graph over years by different countries. India was very back here, but you can see it's a slow and steady increase in the case of India. Its yield and quality value are improving, and you can see a sharp improvement over this period. This is marked. This is mainly attributed to the success of varieties 238. So we are, I think, almost second in the world now. After this pink line is for United States of America. There the value is high because the area occupied is much less compared to India. So in the global level, if you take, we are the leaders. We are going in the right track to our success compared to the area of other countries. Brazil and India are the leaders. And we can see ours is a very steady improvement, except this is the drought period where there was a drop, and slowly we are marching towards excellence. And training programs like this are motivational for a collective action from a group of different disciplines for the successful sugarcane cultivation. Thank you and Jai Hind.
Thank you, ma'am. Thank you for those valuable insights. If there are any questions or queries regarding the topic, please ask.
One thing is that apart from varieties, the different topics will be dealt by experts from SBI as well as from other stations. But anyway, if you have some specific questions, you can.
There is a question in the chat regarding how the numbering is given to cane varieties.
How?
If you can read the chat window, they are asking, one of the Gayatri is asking, ma'am, the presentation was very informative. How numbering is given to cane varieties?
That was I was telling now. For example, 0238, 0238. First two digits are standing for is for denoting the year of identification. That this variety was identified in the year 2002. And rest are the culture number, which we will be having many clones under testing. 38 clone. This is the 38th clone selected during 2002. 11015. 11015 first two digits as a 2000, that is identified in the year 2011. 015 is for the culture number, the 15th clone of that year. Another one, 2673 for example. That is identified in the year 1987, and the rest are the culture numbers. So if you take a typical example of a variety from other station, for example, CO BSI 12121. Okay, so BSI, the BSI stands for Coimbatore, Sorry, VSI is not 1 to 1 to 1 because that is only VSI 1 to 1. No, there's no co there. VSI 1 to 1 to 1. First two digits 12 that is identified by them in 1912. That is 2012 which entered ICRIP, and rest is their slot number. For each ICRIP center, there is a slot number. For VSI, it is starting at 121, 121 to 130. Like that, the varieties are nomenclature. For another variety, for example, COA. If you see the correct name, you will know from which station the variety has come. COA, the A stands for Anakapalli. COC stands for Kadalur. COS stands for that is crossing done at Coimbatore, and the selection at Kadalur. The COS means Shah Jahanpur. Like that, it's a very fixed and very clear thing. Anybody has some experience in sugarcane will be easily able to identify the year as well as the name of the variety, number of the variety. Anything else?
There are further questions in the chat. One of them is like reasons of flowering. The other one is energy fiber cane trays are made.
Fiber cane trays are made. So that is just like you make from pulp, no? Bagasse, sugarcane bagasse. Once the sugarcane juice is extracted, the rest of the material is called bagasse. Bagasse is a very valuable material. Actually, sugarcane is a kalpa vriksha now. Every part of the crop is important. So the bagasse can be pulped, and then from that the paper, that is this particle board, or even this plastic, this disposable culinary or the cutlery can be made. So that's just a very simple technology. And similarly, in other, even this bagasse can be used as a platform for culturing mushrooms. Then bagasse can be used as mainly it is used as a fuel for making jaggery. You know, the juice will be extracted from the cane, and it is used as for burning. So by even bagasse can be converted into ethanol. That is secondary, primary ethanol, secondary ethanol, tertiary ethanol, like that. I think this will be dealt by Dr. Hari and subsequently in the lecture. Then I want to know about the technology. Okay, that will be covered later. Then, madam, how we can reduce the flowering by changing the date? I will tell you, the flowering in sugarcane is during the months of October to December. Sugarcane does not flower before that. So if you plant a short duration variety in the month of January, short duration variety is 8 to 9 months. You harvest in November, you are skipping flowering. October first week, if you harvest first week or second week or November, you are, once it flowers, after 2 months only there will be quality deterioration. So you harvest in October, you take up the planting and the ratoon crop, you harvest in July, and then like that, you can easily skip flowering. So you by adjusting flowering, you plant in November, take up the harvest in December, in August, September. Then you have the ratoon crop, we harvest in March. Then the second ratoon crop in October. Like that, that has to be worked out by knowing the real planting time of a particular region. In some plant areas, October planting is very common. In some plant areas, July planting is common. So you have to just design in such a way that October, November does not coincide with the maturity phase of sugarcane. That way you can prevent flowering.
Any other question? In Muzaffarnagar area of UP, of wild pig problem is there. How can we control it? At least last 4 years we are working in farmers.
Not only in Muzaffarpur, even in our breeding farm at ECC, we have wild bird problem. So that is actually the work of an entomologist. So recently we have installed one machine, that is one that is called some sound alarm that is mimicking the sound of several wild animals. So if you play that, it is said that the wild boars are scared, but those are some of the ways by which you can prevent wild boar. But my concern is that some of the varieties are chosen specifically wild boar, especially soft varieties. So by slightly increasing the fiber content, this becomes a less choice for the wild boars. So now the present fiber content is around 12%. If you increase by one unit or 1.5 unit, 13% or 13.5%, it becomes a little hard for the wild boar not to destroy the crop. That is that is a breeding way of controlling wild boar, but otherwise it is an entomological and some other method of controlling wild boars.
I think we have gone way past our time. Thank you, ma'am, on behalf of NADCL and on behalf of the organizing committee. I would like to thank you very much, and even on behalf of the participants. Thank you very much for being with us. Thanks a lot, ma'am.
Thank you very much. We will interact again. If you have any specific questions, no, you can mail to me, or you can give to the NADCL. They will transfer the questions to me. I can answer subsequently, because I will be there with you for 21 days. Thank you very much, and wish you all the very best during this training program. Thank you.
Thank you. Thank you.
Thank you, ma'am. Thank you, ma'am.
Thank you. Thanks, everyone. Thanks for joining. That's all for today, and we'll be joining back again tomorrow at 11. Links will be shared on your WhatsApp group. Thank you.
Thank you. Thank you.